Good afternoon, and welcome to the ISCB Fellows Keynote presentation. My name is Paul Horton, and it's a deep honor to be able to introduce our keynote speaker, Gary Stormo. Gary Stormo began his college education as an undergraduate in biology at the California Institute of Technology. He then did his graduate work and his early faculty career at the University of Colorado at Boulder. Since 1999, Gary has been at the Washington University Medical School in St. Louis. Gary is a prolific writer and author. According to Google Scholar, his papers have been cited a total of over 18,000 times with an H-index of 66. In fact, he has over 50 papers which have been cited at least 100 times each. And if you look down the list, the majority of these are 2, 3, 4 author papers in which it is evident that Gary has directly contributed to the scientific achievement. Gary has pioneered several important problems in our field, including motif discovery, gene expression regulation analysis, RNA structure prediction, and as I learned earlier today, direct coupling analysis. And I trust that Gary's going to tell us about some of these problems this afternoon in his keynote address. Therefore, I will leave that to Gary and instead briefly relate a couple of events which give me a personal feeling of gratitude towards Gary. The first event was, in fact, the very first scientific paper I remember reading. It was entitled Identification of Consensus Patterns in Unaligned DNA Sequences Known to be Functionally Related by Gerald Hertz, George Hartzell, who I later became friends with at Berkeley, and Gary Stormo. This was published in 1990 in Cabios. I don't know how many people here remember Cabios, but that was the predecessor of what we now call the journal Bioinformatics. This was a seminal paper in motif discovery. It fascinated me and many other people, and in fact, I later did some work in this area. The second event was a bioinformatics workshop organized by Gary Stormo, Christian Burks, and Eugene Myers, held at the Aspen Center for Physics in the summer of 1994. At least that was the year that I attended. This was a three-week workshop with only 23 participants. Amongst the participants were a few unknown graduate students, including myself, the 3 organizers I just mentioned, and in addition to that, the likes of Tim Bailey, Sean Eddy, Tony Kazak, Rick Lathrop, Susanna Lewis, and Bill Pearson of FastA fame. You can imagine what a formative experience it was for a graduate student to be able to spend 3 weeks in close contact with such accomplished scientists. So Gary had a very large influence on my career, and in fact, in numerous scientists' careers. In the interest of time, I'll just mention 3 names. The first is Tim Bailey, very well known for the motif discovery program MEME. I have direct from Tim that he feels he would not be where he is today without Gary, and I learned that in the very early days of MEME, he called Gary from San Diego to Colorado, not knowing Gary, but they had a very deep conversation on the subject for approximately 1 hour, and I'm sure this greatly influenced the development of MEME. 2 more names, which I'm sure everyone in this room is familiar with: David Haussler and Eugene Myers. These 2 giants in our field were originally purely computational scientists, but they happened to be at the University of Colorado at the same time as Gary Stormo, and the 3 of them regularly met to discuss science. It is easy to imagine that without the biological insights that Gary Stormo brought to those conversations, that David Haussler and Eugene Myers may not have discovered the challenge and the beauty of computational biology, and imagine what our field would be like without their contributions. So, in summary, Gary Stormo has contributed immensely to our field in 2 ways. Indirectly, through the profound influence he has had on the careers of many scientists, and equally importantly, directly through his large body of research, which I'm sure we're all dying to hear more about. So without further ado, I would like to yield the podium to Professor Gary Stormo to tell us about searching for signals in sequences.
Thank you. Is my microphone on? Okay. Thank you for that very kind introduction, Paul. I sort of feel like I should stop now and take questions rather than lead to a disappointment. Anyway, I picked a title for my talk which sort of covers a lot of things I've done in my career. They asked me for a title a couple months ago, and I sort of decided yesterday exactly what I was going to talk about, so it left me a lot of flexibility. And I can empathize very much with Gonzalo, who yesterday talked about the difficulty in deciding what to talk about for a meeting like this. I don't want to just give my sort of most recent research. I want to give a little bit of background and so on. So I started making a list of things that I like to talk about, and another list of things that I know a lot about, but unfortunately the overlap isn't very big. And in fact, this is an exaggeration. It's probably more like that. But nonetheless, I came up with a plan. What I'm going to do is I'm going to give you a little bit of history. And this is a bit of history about the field in general, but mostly related to my own interests and what I've been involved with. I'm going to try to bring you up to date on our current research, and I hope actually to talk a little bit about some unpublished work, squeeze it in there. And then what I want to do at the end is actually go into speculation. So this is something that I don't know a lot about. I mean, you know, I can't predict the future any better than anybody else can. But think about sort of where the field is going. Sometimes students come and ask me, you know, what direction do I recommend that they go into? And so I've thought about it a little bit where I think the field's headed. So I'm going to spend a little bit of time, I hope, if I haven't picked out too many slides. But like I said, you know, thinking about the future is, of course, hard. As Niels Bohr said, prediction is very difficult, especially about the future. Now, I would argue that actually this should be amended, because I think if you turn on the TV almost any time, you can find a whole bunch of talking heads, you know, predicting about the future, never agreeing with each other. But so it's really only its accurate prediction that is very difficult, especially about the future. And I want to add one more quote. This is from a famous American philosopher, Yogi Berra, who said, the future ain't what it used to be. And if you think about it, I mean, this is the most concise, best definition I've ever heard of what it is to be a Bayesian, I think, right? So, yeah, I mean, who would have thought? Okay, so let me jump right in. Oh, I should say, when I did put all this talk together, I had a lot of slides, so I took a bunch out, and in particular, I took out all the rest of the jokes. So you're just going to have to use your imagination for how funny this talk would have been if I hadn't taken all those out. Okay, so here's a bit of the history. I'm going to talk about the basic model that we invented a long time ago, and it has sort of become a fundamental, the position weight matrix model, also known simply as a weight matrix or position-specific scoring matrix. And I'm sure for this audience, you know what it's about. Although, it's interesting, I was out looking at some of the textbooks out in the lobby, and people don't necessarily agree on what a PWM is. So I want to spend just a minute telling you what my definition is. So the idea is you have a matrix. In this case, you have a score. Just think of that abstractly for the moment, a score for all possible bases, A, C, G, or T, at every position in some binding site. And so then given any sequence, you can calculate the score for that particular sequence just by summing the numbers that correspond to the sequence. So this gets a total score of 24 because it's got a C in the first position and a T in the second and so on. Okay, and you can think of then just taking the matrix and sliding it along and you get a score for the next position. And this would be the consensus sequence because it has the highest value in every position. So in my mind, what a PWM is, it's an additive model. You can think about encoding the sequence itself as ones and zeros, where you have a 1 for which base occurs at each position. And so if you think about it that way, then it's just the dot product between the matrix and the sequence. So it's a linear model, it's additive. The SI, that encodes the sequence, and the W weights the different bases at each position in the sequence to give you a score, which hopefully is related somehow to functional activity. And if you think about it this way, then it's easy to extend the model to include additional features. So one of the fundamental things about this model, and one of the limitations of it, is it assumes the positions are independent. If I get a score for an A at a certain position, it doesn't matter what its neighbors are. And that may or may not be the way proteins work. As you see later, it actually, I think, is a bit surprising. It works pretty well a large fraction of the time, but not always. So you can add more things. If the positions are not independent, I can include a dinucleotide feature or something like that. So whatever I think is important, maybe I'm going to add features having to do with the structure of the DNA in addition to just the sequence. Okay, so then the real question is, where do the numbers come from? How are the parameters estimated? And over time, there have been a variety of methods that have been published to do this. The one I talked about that we first did was a discriminant learning method, where we had a collection of ribosome binding sites, and we had a collection of AUGs that weren't ribosome binding sites, and we simply found a weight matrix that would distinguish the two sets. After that, soon after that, came some probabilistic modeling, and I'm going to describe that in a little more detail in a second. A few years later, we realized, and other people as well, that in fact, if you have quantitative data, so the first things I talked about is we just had a collection of sites, and maybe a collection of sites and non-sites. But if you actually had quantitative data, and you knew that this site had a certain affinity, and this site had a less affinity, and so on, you could actually just apply regression to it. So you could identify, my brother's online, good. It's pretty early in California. And I want to specifically emphasize binding energy models. These different classes are not necessarily mutually exclusive, but I want to emphasize a little bit binding energy models because they have some characteristics that set them apart. And in a binding energy model, the elements of the matrix are actually the energetic contribution of each base at each position to the total binding energy. So this is the probabilistic model. Basic idea. This is an old slide, but the idea is pretty straightforward, which is that I take a collection of binding sites. So this is -10 data from E. coli promoters from a long time ago. At the time, I think we had 242 promoters. So you can line them up. You can just make a count, the number of each base at each position. That's the raw data matrix. And just turn that into a frequency matrix, the frequency of each base at each position. Now, when I said there seems to be a disagreement, some people treat this as if it was a PWM, but to me, that's not a PWM because it's not an additive function. If I want to calculate the probability of a sequence being in the -10, I would multiply those probabilities together, not add them. Some people do add them, which is don't get me started. But that's a probabilistic model, and in fact, that was first published in 1983 by Haar et al. On E. coli promoters, and it was really a hidden Markov model. It even included a variable spacing parameter because the distance between the -10 and the -35 was variable. And so, it was a really early version of that kind of thing. But then a year later, Staden turned it into a real PWM because he just took the logarithms. So you turn a multiplication problem into an addition problem by taking the logarithms. And then we added one more feature, and we said, well, what we really care about is the relative frequency of the observed set compared to what you expect by chance. So in this case, PB in the denominator refers to the background probability of each base. And the idea is here, if that's a constant, like it is in E. coli, you could sort of ignore it, it's just a constant. But in some species, that's not true. Yeast is about 2/3 AT, and some other species that we've worked on are 2/3 GC. So what we really want to know is, how does that observed frequency, given that you're in part of a collection of binding sites, compare to what you expect by chance? And so our definition of the weight matrix was this log odds method, okay? And you can take that one step further, and you can calculate what we call the information content. And there are a variety of ways to write this formula. I think the easiest way to think about it is, here it's shown for each position independently, is it's the dot product of these two columns. Or another way to think about it is just the average score of all the examples in your set. Now, information content is, in computer science, it's called relative entropy. In statistics, it's called Kullback-Leibler distance. It's a likelihood ratio statistic. And in fact, it's very often used in motif discovery methods because it's a nice measure of statistical significance. And in fact, you're probably familiar with this. So this is a method, sequence logos, invented by Tom Schneider, who's also the one who pioneered the information content analysis. And this was published in 1990, and I think most of you are familiar with this. There are other ways to do logos, but this is sort of the classic method, where at each position, you plot the entire information content at that position, and then the individual bases are in proportion to their probabilities. I put a note here to remind myself, I just wrote a review on sort of the history of this sort of stuff if people are interested in it. It should be out at any time. It turned out, I didn't write a review from the very beginning till now, they wanted a review of about the first 10 years, sort of the early history of this. And it turned out that was really easy because between 1982, when we first our first paper, and 1990, there were only 10 papers published. And half of them were from my group. So it was actually an easy review to write. But it was also, I mean, these are papers that were methods papers, I mean, how to build. There were a few other papers that actually used position weight matrices, you know, to make predictions and so on, but actually very few. Most people in those days used consensus sequences. And it was kind of, you know, frustrating for us because we'd shown, in fact, that a weight matrix is at worst equal to a consensus sequence. You can make a weight matrix that's actually exactly equivalent to a consensus sequence. So then you can say, well, in the worst case, it'll be no better than a consensus sequence, but it could be quite a bit better. So, so we were disappointed that the method hadn't really caught on. But from a biologist perspective, I mean, when they write the method section, it's easy to say, well, we searched for TAT, AAT, and we allowed 2 mismatches, kind of stuff like that. Whereas writing a matrix of numbers and stuff, it was sort of hard for them to, for a lot of people anyway, to come across. So it didn't get used very much until Tom invented logos. Now they could say, we searched for the Tata box motif, see figure 2. And even if they didn't, you know, really know exactly what was going on underneath, it made a very intuitive view for it. So suddenly, starting in about 1990, after this paper came out, people started using weight matrices a lot more often. And in fact, this paper, which didn't introduce any new mathematics or methodology, but it introduced a new visualization tool, is more cited than all of the previous papers on weight matrices. And it just sort of, I think, points out how important it is to get your point across, and sometimes in a nice visual way. There other things were happening also that made weight matrices more useful. And one was that we're getting new kinds of data. So the whole Motif Discovery process started in, we wrote a paper in, oops, do I want to go there yet? Where am I? Motif discovery process started around this time, where now in the early work, we actually knew what the sites were, and we were just trying to come up with a model that would let us search for new examples. Later it came where we didn't really know where the site was. We could say, well, this promoter must contain a site, and this promoter must contain a site, so we do motif discovery. So that also started around, well, our first paper was in 1989, and then in 1990 there was the EM algorithm, and a few years later, MEME and Gibbs sampling. So that really took off as well. So that was another reason that motifs became more popular. So in this slide, I just want to point out that, sorry, I went too far. This slide, the log odds method that I just described, I mentioned I wanted to bring up energy models. Well, under some conditions, they're equivalent. And von Hippel first pointed this out, Berg and von Hippel. If the binding, so here's the Boltzmann equation, right? This is the posterior, this is the frequency of a site being bound. This is the prior, you know, the overall probability of seeing that sequence in my collection. And this is the energy for that sequence I. So this is just the Boltzmann relationship. If it's a non-specific binding, so the energy is the same, then the prior and the posterior are equivalent. Otherwise, you know, the posterior will be different because you're selecting things according to this Boltzmann distribution. So I just rearranged this and I get the same equation I showed you before, except there's a minus sign here. Biologists like the good guys to have a positive sign, and of course physicists know that they have lower energy, so you just have to think about switching the sign. But anyway, so under that assumption, then the log odds method and the energy method are the same. However, we knew that was going to be an approximation, and this paper really drove it home by Georgievich et al. The real equation is this, the so-called Fermi-Dirac equation, where we have a new parameter which has to do with the protein concentration. And as you raise the protein concentration, of course, the probability of everybody is going to go higher, but you can't go above 1, so you saturate. So what's shown in these two graphs is, this is the Boltzmann one, where now, for the consensus sequence, which we define to have energy 0, we define its probability to be 1 because we're showing the relative probability. That is, it drops off exponentially as you increase energy. Over here, I'm showing you the absolute probability, which can't go above 1. And this would be a case where the consensus sequence is about, say, 95% bound. And then it drops off more slowly, obviously. So these two curves look quite a bit different. And it actually has an effect on how you make predictions. So for instance, let's suppose I had a mutation that increased the binding energy by 2 kT. In the context of the consensus sequence, it would go from maybe 95% bound to 80% bound, a small change, a 15% change in binding affinity. in the context of something that was, started out about 50% bound, it would drop way down to 10%, a five-fold change in binding affinity. So it would appear that that mutation was in fact context dependent. But I told you already, it's an additive. Energetically, it's additive. You can show that. I can make a few examples. I'm just going to go over this quickly. This is a protein where we actually went in and measured all the binding energies to all the single base pair changes. Here I'm showing the consensus sequence with 0. Here I've rescaled things. This is minus energy, so the good guys are on top, and I'm actually showing the different energies where I've now set the average to be 0. And if you do just a calculation or a simulation at low protein concentration, you get this logo and this frequency matrix. At high protein concentration, you get this one and this one. And you could say, well, it just looks like it's less specific, it's kind of scrunched down. But really what's happening is the probabilities are, when I go from low to high concentration, is they're moving toward the mean. So some of them go up in probability and some of them go down in probability. They all go toward 25%. And the effect is that, in fact, what happens is when you make predictions, sometimes the order gets inverted. This is the true order. The Fermi-Dirac is monotonic. It doesn't actually change the order of anything. But what happens is the predictions can change, and that's because you can no longer factor it in independent probabilities, even though it's additive in energy. So it's for these reasons that we've really sort of focused on energy models, because we think they really focus in on something, whether something is additive or not, independent of whether the frequencies will lead you to that same conclusion. So I want to talk just a bit about some high throughput methods, and I want to first point out that what we care about is specificity. If I have one sequence, here's my transcription factor of interest. If I have one sequence, classically you would determine its affinity, its KD, by just measuring probability of binding as a function of concentration. But what we really care about is we have a whole collection of sequences, and we want to know how the affinity changes as a function of the sequence. So we really like to have this kind of data for every sequence. And there are methods like Matomi that let you do hundreds at a time. But in fact, the high throughput methods I'm going to talk about, what we really do is we take a slice through the data in this dimension right here. So we have some protein concentration. We may not even know what it is. But what we measure is the probability of essentially the entire set of sequences at once. And then we fit it to the model and estimate what the energy parameters are. So I'm going to talk very quickly about 3 different kinds of high throughput data. And I'll go into each one of these separately. But the idea is that we collect data in high throughput manner. We wrote a review a couple years ago about the experimental methods, and then we have some modeling software that lets us determine what the specificity is. So we called it high throughut CellX. I think CellX-Seq is actually a better name, and that's what most people I think call it now. CellX is an old method. It was actually invented by my thesis advisor, but I wasn't involved in it at that time. And it was originally designed to select for RNAs that would bind to proteins, but most people have actually adapted it to DNA binding proteins. So you start with a random pool. It could be synthetic DNA or it could be a chopped up genome or something. But anyway, it's a random pool. You have a way of isolating the DNA that binds to your protein. So in this case, the protein is immobilized on a column or something. You wash it through. Some of the DNA is unbound and you just throw that away. And you collect the bound DNA. Now in the old days, because there's always a lot of background, some of the DNA will bind to the protein non-specifically. Some of it will just bind to the column. We can't really distinguish those two. But there's always a lot of background. So what they would do is they would amplify this by PCR, do several rounds, and then clone and sequence 20 or 50 guys. And you would learn a lot about the specificity. You would learn what the consensus was and what positions were more tolerant of variability and so on. But it wasn't actually very quantitative. So what we do now is we take the random pool, we sequence that, so we actually know how random it is to start with, and then we sequence the bound stuff after just one round. And the data look like this. So here's this big peak right here of sequences that just showed up once. We only did about 500,000 reads in this particular experiment, so we didn't even sample the entire, this is a 10-long randomized sequence. But anyway, there's a large collection of sequences, and that's the background. But then if you blow this up, there are also sequences that occur hundreds of times, and that's sort of the information that we're going to use to estimate the model. So the math is over here. And I apologize, this little s here is a new, that stands for the state of the DNA, which can either be bound, 1, or 0. But I said we throw away the unbound stuff, so we're only dealing with the bound stuff. So the data we have in our hand is the probability of seeing a particular sequence given that it's been bound. And I said we also sequence the prior, so we know the starting probabilities. We have that probability and that probability. What we want is this one, because this is the one, the probability of binding given sequence, because that's the one that depends on the energy parameters and depends on the protein concentration. And I just show here that we can actually take the total binding energy and divide it into two modes, a specific mode and a non-specific mode. So we end up with about 30, this is a 10 long binding site, so there are a total of 32 parameters. 30 for the 10 positions of the binding site, a non-specific parameter, and a protein parameter. And basically, we just use Bayes' rule to take the information that we have and use it to calculate the information that we want. So we basically do a nonlinear regression. We get a very nice fit to the data. This is showing a comparison with Bioprospector, which is sort of an off-the-shelf Motif Discovery tool. We actually tried several ones. Bioprospector was the only one that didn't crash on the amount of data we gave it. Now, that was several years ago. I think nowadays there are probably other programs that would also handle that kind of data. But I don't think they would do any better if they have, because they have the same sort of underlying model, essentially this Boltzmann assumption, which isn't really quite valid. So anyway, we get models that fit the data quite well. So let me jump to, I didn't mention the name of the, so the program we called BMO, which stands for Binding Energy Estimates by Maximum Likelihood, written by my student, Yui Zhao. So then we looked at protein binding microarray. The data is kind of different now. I'm sure you're all familiar with microarrays, except these are double-stranded microarrays, double-stranded DNA, and they're designed in such a way that they contain every 10-long sequence somewhere on the array, one time on the array. And now what you do is you add your protein, your transcription factor of interest, and you have a way of detecting it, some fluorescent label. So it's like a microarray of the old, except what you're actually measuring is protein binding to DNA. The data look similar. We can take experiments, and we can often get good motifs that give you a good fit to the data, but not always. Here's an example where, and now I'm showing you a different kind of logo. This is the energy logo again. This is one in which we didn't get a particularly good fit to the data. R squared is 0.58. And so this is a case where we went to say, well, maybe we need a more complicated model. So Yui actually looked at adding dinucleotides all across the whole site. It turned out that only one dinucleotide really mattered. Oops, wrong direction. If we included a dinucleotide between positions 4 and 5, the R squared went up to 0.83, which was about as good as we did with much more complicated models. And the way to read these logos, I know that's hard to see there, but it's the same as the original weight matrix. You just add up the values that correspond to the sequence. So whatever the sequence is, I would add up the energy values of that, and then I would also add in the value for that dinucleotide, because what we've done here, what I'm showing you is essentially the residual. It's the energy that wasn't captured by the mononucleotide model. And we have a way to do that very efficiently. So basically you do one pass through the data and you learn all the independent parameters, and then you fit the residuals for all the non-independent parameters. And we did this for the whole data set, the 400 some examples that were in the database from Martha Bullock's lab. And we concluded that almost every protein could be fit pretty well by one simple PWM, which conflicted with some earlier reports. But our result has recently been corroborated by the same people who did the original report. In this paper, which just came out a couple months ago, they compared 26 different algorithms on 66 different factors, in which for each factor they had 2 independent arrays, and so they could train on one and test on the other. And the conclusion was that about 90% of the transcription factors could be fit quite well by a single PWM. There was some for which you did better if you included dinucleotides for at least some of the positions. And there were a few that actually required multiple modes of interaction. And in fact, there's a really nice paper from Martha Bullock on one of those examples that just came out 2 weeks ago. So there are cases where you need to fit multiple models, but it's actually, so far at least, appears to be a relatively small minority of factors. And it's, I think, somewhat surprising that actually PWMs do as well as they do on most of the data sets. Furthermore, they compared the models that came out of this with actually ChIP-seq data. And what they found was that you could predict the ChIP-seq data as well as if you had learned the motif from the ChIP-seq data itself. So that really tells you that you're learning the intrinsic specificity quite well for these PWMs with PBM data. I'll talk about one more high-throughput method, bacterial 1 hybrids. So this is not really an in vitro method because it's done in E. coli, but we treat it because you can take any transcription factor from anywhere, and we just sort of treat the E. coli as if it was the test tube. The advantage of this method, in my mind at least, and I think people agree over the other 2 methods I talked about, is you don't have to purify the protein. all you have to be able to do is express it in E. coli. And most proteins you can express in E. coli. So you take whatever transcription factor of interest that you have, you fuse it to omega, which is a non-essential subunit of RNA polymerase. But what that does is it takes any old protein whatsoever, any old DNA binding protein, and turns it into an activator because it has an affinity for RNA polymerase now. Then you have a random library of binding sites, and the cells are set up in such they don't grow. You don't give them histidine, so they need to make this enzyme in order to grow fast. And so you have a selection for things that have good binding sites. It has a good binding site, it makes a lot of this thing, and it grows fast. In the old days, this was a thing where they would plate them out and they would pick the top biggest 20 colonies or something and sequence them, and they would learn something about the specificity. But now what we do is we just scrape the plate, send it off for Illumina sequencing. Again, you get this background because the cells that didn't grow are still on the plate. They just didn't grow, so they show up with a very low frequency. But again, you get this long tail of sequences where cells are growing very fast, so they give rise to big colonies. But we just sequence it all at once, and again, we go through and fit doing a nonlinear regression method. It's a little bit different mathematics now, and the reason is because, remember, these are on plates, and so the number of counts of a particular thing is not directly related to the affinity. It's related to how fast the cell grows, right? And so if you let the cells grow longer, you'll get more of those counts. So what you have to do is actually compute the growth rate, and we do that by, we sequence the original library, so we know the number of each sequence I at time 0. We let them grow for a certain amount of time, and then we count how many occurrences there are of that sequence at time t, and that lets us determine what the growth rate is for sequence I. And then the assumption we make is that the growth rate is related to the probability of binding. The better you bind, the faster the growth rate is going to be. And that's going to be true up to some level at which you saturate, and histidine is no longer limiting. So the equation is a little bit different, but it's basically, here's the Fermi-Dirac part, and now we have a maximum growth rate for what happens at saturation, but now we're fitting to the logarithm of that ratio. The data look like this. It's a little noisier than the in vitro experiments, but we can still get pretty good models out of it. This is just a slide I showed earlier. We have different kinds of high-throughput data. We have algorithms that can take that data and generate models that, for the most part, tend to give us pretty accurate specificities. So what are we going to do with that? Well, there's lots of things you can do with it. Of course, you can use it to try to predict binding sites in vivo. Now, if you just try to predict binding sites in vivo across the whole genome, most of those are false positives because the regions simply aren't accessible. But if you combine them with things like DNase 1 sensitivity, so you know which regions are accessible, and then you ask which motifs match in those things, it actually can be an effective way. It's been shown in a couple papers from the ENCODE project that that's actually a good way to identify which factors are binding to which enhancers and so on. And it's much faster than doing a ChIP-seq, which you have to do one at a time for every single factor. Here you do one DNase 1, and then you have a library of motifs, and you try to match which motifs bind. Now, of course, we don't have motifs for every factor yet, but anyway, you can make good progress doing that kind of a strategy. There are other things you can do, but what I want to talk about just for the next few minutes is an age-old problem. It used to be called the recognition code problem. You would think that if you had a DNA binding protein, and you know structurally how it binds to DNA, and what amino acids are interacting with the DNA, you ought to be able to predict what its binding site would be. But it's been a problem that's plagued people for a long time. There's a famous paper actually by crystallographer Brian Matthews, the title of which, this was published in Nature, and the title of which is "There is no recognition code." Now, what he was talking about, and he was very clear about this, is a very specific idea of a code, a deterministic one-to-one code, and that clearly is not the case. So we started working on this problem, and we defined them as being a probabilistic code. And the first time we submitted this grant, it got turned down, and the reviewer said, well, everybody knows there is no code, and so, So then we changed the name to a probabilistic model instead of a code, and then it got funded. So anyway, so what we're going to do is we're going to take data sets where we have several proteins and we've measured their specificity. We're going to infer the interacting positions by looking at covariation between the protein and the DNA. And then we're going to apply machine learning methods, support vector machines, random forest, k-nearest neighbors, neural nets. We tried several different methods, and we're going to assess how well they do by cross-validation. I'm going to show you results from two families, zinc finger families and homeodomains, which are the two largest families in most eukaryotes. The data set for the homeodomain family is a mixture of our own data, B1H data mostly from Scott Wolf and Michael Brodsky, and also published data from Tim Hughes and Martha Bullock, mostly from mouse. We had a total of 264 independent homeodomain proteins for which we had determined their specificity experimentally. Here's that. If anybody would like to order this as wallpaper for your bathroom or something, just let me know. I think it would look quite nice, actually. Anyway, that's the data that we had. So we identified motifs, and we could align them, actually, we think, quite reliably. Here's the covariation analysis. So we have the protein logo, so our 264 proteins, our 264 binding sites. Now, I should say that for this particular data set, and it actually is true in general, this N at position, 51 there, the asparagine there, is absolutely conserved, or almost absolutely conserved, and so is this A, and they interact with each other. So we don't learn anything about that, right? There's no covariation because there's no variation. But other than that, the things that show up as the highest covariation are marked with circles here, as the letters here, and they end up being the positions that, in fact, are known from crystallography to interact with the protein, do the specific, with one exception. Position 19 is not known from crystallography to interact, yet it showed up as one of our high covariation analysis. So this is how we evaluate how well the methods do. What we're going to do is we're going to add in positions, and they're put in the order based on their covariation analysis. We're going to take initially a single position of the protein and say, try to predict the entire motif. Well, obviously it doesn't do very well. So the axis here, the y-axis, is the mean squared error. So we have an experimentally determined probability matrix, and we have a predicted probability matrix, and we're just computing the difference between them, the squared error of their differences. And of course, obviously, like I said, if you just take one position, you don't do very well. You add two positions, you do a lot better. By the time you get to 7 positions, you've done quite a bit better. I'm showing you here data for random forest and support vector machine, which in our case worked essentially the same. The random forest was a little bit faster and easier to train, so we ended up focusing most of our effort on the random forest. What I'm showing here in blue is the k-nearest neighbor method, which was the best method that had been previously published. So we're showing that we can actually reduce the error by quite a bit over the previously best method. So what does that mean? An error of about 0.008 or so. So I just show you several examples. So these are all examples that were picked to be around the mean. That is, half of the things we do better than this, and half of the ones we do worse. So these are all about the ones that do about average. What I'm showing you here is the actually experimentally determined motif and our predicted motif. In every case, we get the consensus sequence right. In some cases, we're pretty good on the quantitation. In other cases, the quantitation's not quite so good. But anyway, this sort of gives you an idea of how well we can predict a homeodomain protein specificity if you just give it the protein sequence. And there's a website where you can do that. We've now done the same thing I mentioned for zinc finger proteins. This actually, we're in the process, so we haven't published this yet. We're in the process of writing up the paper, and this particular figure is out of date. This, I think, is 250 factors. We now have 650, so about 2.5 times as much data as this. I'm not sure I could make a figure with all those on it. We did the same thing, random forest prediction. We ended up with similar kinds of overall squared errors, and this is just showing you again. I picked a few examples that are all near the mean. So in other words, half of the time we do better than this, and half of the time we do worse than this. But I wanted to pick out a bunch of examples where showing you the diversity of zinc finger proteins. Whereas homeodomain proteins, the motifs are all actually pretty similar to each other. For zinc fingers, you can have ones that are GC rich or all A's or TA's. You can have a whole variety of different motifs that you can select zinc fingers for. And so again, once again, we think we're doing pretty well with these particular predictions. So summary of this part of my talk is PWMs can be good representations of specificity for a large fraction of transcription factors. Extended PWMs, so you add additional features, for instance, dinucleotides, if the positions appear not to be independent, can be used if they help. Regression methods, I think, are really the way to go if you have quantitative data, because first of all, it tells you how good is your fit, and do I really benefit or not by adding additional parameters. And I mentioned just a little briefly why I think energy models have some advantage over the classic sort of simple probabilistic models. You can make more complicated probabilistic models that are essentially equivalent to the energy models, but that's not the way they're usually done. So now I'm going to jump to the second part of my talk, which is about speculations. And so I'm getting away from sort of what I've spent most of my career on, although actually in the early days, most of you I'm sure don't know this, I'm going to show you a couple examples. I actually worked mostly on post-transcriptional regulation, and we studied several different proteins that were regulated at the level of translation. And what I'm speculating about here is that in fact, when things are all worked out somewhere down in the future, what we'll have found is that there's more regulation happening post-transcriptionally than transcriptionally. I don't know if that you consider that a surprise or not. Most people would say, well, isn't every gene regulated transcriptionally? You know, maybe there's some housekeeping that are always on, but you could say, okay, every gene is regulated transcriptionally. But I would say a large fraction of genes are going to be regulated post-transcriptionally, but in addition, they can be regulated in lots of different ways. You can certainly affect their translation. You can also affect splicing. Now, well, you might say, well, splicing is not really post-transcriptional, it's co-transcriptional. But when you think about how people draw regulatory networks, they're always talking about how you control the initiation of transcription. All these factors come together, and all these regulatory networks are all about the initiation of transcription. So maybe I should call this post-transcriptional initiation regulation. And then I get to include regulation of splicing in that category. So you can regulate that. You can regulate the half-life of the RNA. You can't do that to the DNA, right? You can regulate where it's localized, and that can affect, you know, what its functions are, what activities it participates in, et cetera. You can do editing, which we're just appreciating is more common than we used to think. You can do frame shifting. There are a variety of things that you can do post-transcriptionally that don't happen at the DNA level. And my guess is that when we, and part of the reason that, you know, I said when people come and ask me what they should work on, you know, young students, I said, you know, there's a lot we don't know about transcriptional regulation. There's a lot to be learned still. But I think we know 10 times more about that than we do about post-transcriptional regulation. And when it's all done, there's going to be at least as much post-transcriptional. So there's a big opportunity there to get in on something early that's really going to blossom a lot. That's my prediction anyway. Also, another speculation is that autoregulation, post-transcriptional autoregulation is very common. Now, there are many sort of classic examples. I'm going to point out a few. But my guess is, and again, this is speculation or guessing, is that it's actually much more common than we appreciate. And I'll show you why I think that as we go on. One of the reasons is that any protein can bind to RNA. Now, there are, you know, classic RNA binding domains that people use to predict RNA binding proteins. And those are ones that, you know, they bind to some motif, and they probably have lots of copies of that motif, and they're really involved in that. But if you think about it, I mean, the original Cell-X experiment that I talked about early was you take a protein, then you give it a random pool of RNA, and for every protein, you could find an RNA that bound to it. So in that sense, finding a protein that doesn't bind RNA is equivalent to finding a protein that doesn't bind antibodies, right? Now, that doesn't mean that's what it normally does inside the cell. That's probably a small subset, but it certainly has the capability of binding RNA. And so given that capability, and given that evolution is very opportunistic, my prediction is there'll be lots of cases where proteins, as a sidelight of what they normally do, also bind RNA and can use that to control expression. I'm going to show you some examples that led me down this speculation. So just think about the overall process of, you know, this is just the central dogma here, transcription, translation, and every one of these steps can be regulated. I call this pre-transcriptional regulation. That is sort of setting up the DNA in order for transcription to happen. That includes things like chromatin modifications, epigenetics, nuclear architecture, all that kind of stuff. Now, you could argue, well, that's also sort of co-transcriptional. That gets set up in conjunction with enhancers and things that are active. But I could sort of set it aside and say that sort of happens prior or maybe co. But then you have the transcriptional regulation, which involves TNA transcription factors binding, and they can be activators, they can be repressors. You have modules of multiple proteins coming together. You know, you have looping interactions, and then you have the whole signal transduction cascade. You have a whole bunch of things that all focus down. But in the end, the result is the gene is on or off, or maybe it's up or down. You know, you can modulate its expression over some intermediate range. Okay, so there's a whole bunch of things that come together to control the transcription. And this list is sort of things I just mentioned a minute ago, but I'll just go into a little more detail. So post-transcriptional, at least post-transcription initiation, you can also modulate translation. So you can do the same thing of turning genes on and off or up and down at that level. You can alter how they're spliced, so what products they make. You can modulate their stability, modulate their localization. You can do editing, you can do frame shifting. You can do all those kinds of things post-transcriptionally. And we know lots of examples of those, but my guess is that we are just at the very tip of the iceberg, and there's a huge amount that we don't know yet. The mechanisms, there are a whole variety of mechanisms, RNA interactions. MicroRNAs weren't discovered so long ago, but already there's a huge body of literature about that. But again, I think there's still a whole lot more that we don't know. Protein RNA interactions, I talked about all these RNA binding proteins. If you actually look at genome sequences and you predict how many transcription factors there are and how many RNA binding proteins there are, the numbers are pretty comparable. And many of those proteins, there'll be splicing factors and all kinds of other things, but they're involved in some of these regulatory processes. RNA can actually do self-regulation. The discovery of riboswitches just about 10 years ago now was a big deal, and I think it is really interesting. And it's mostly they've been found to be highly ubiquitous in bacteria, but they're also being discovered in eukaryotes. And my guess is there are a lot more to be discovered, and they just haven't been found yet. Often these kind of things involve RNA secondary structure, which means that the motifs are much harder to find than DNA motifs are. I'm going to just give you a couple of examples. Here's a paper that just came out 2 weeks ago from Tim Hughes' group, where they took, 205 RNA binding proteins, and they applied this sort of in vitro motif discovery method, sort of like PBMs, but different. They discovered a whole bunch of motifs. Here's some, but these are all sequence motifs. And as I said, many of these proteins actually bind to structure motifs. So for many of them, they don't know what the motif is, or maybe they know part of it, but they don't know all of it. And this 205 is, again, just the tip. It's maybe 10% or something like that of the repertoire of RNA binding proteins. So this is something that's clearly being studied, but I'd say we're way behind in this compared to transcriptional regulation. Riboswitches, I mentioned briefly. This is just from Ron Breaker's website. A huge number of them. They're all over bacteria, lots of different classes, what they recognize. Just recently there was a review in Cell that talked about a decade of riboswitches. So the work, because they really just discovered 10 years ago. And here's an example, again, from Ron Breaker's lab of one in a eukaryote that actually controls alternative splicing. So my guess is we've just, again, tipped at the tip of the iceberg, and there are tons and tons of things we don't know about this kind of regulation. So I want to just give you so I have actually mostly I've worked on transcription and DNA motifs, but I have spent a little bit of time working on RNA thing, and I want to talk to you about one paper that one of my students did, trying to find common RNAs by using a phylogenetic comparison method. So the idea is to find conserved stems. So what we do is we start with, this is just an example where I had 2 sequences, and for each sequence I can compute a probabilistic base pairing model. I think we just used the Vienna RNA package to do this. We get a probabilistic model of the folding of one sequence and also of the other sequence. We also have a probabilistic alignment. And what we're trying to do is we're going to take these probabilistic foldings and alignments and try to figure out what the actual structure is. And we do that by a sampling method. It's not very well described here. This just shows the result after it's gone through multiple steps and converges. You actually get the correct RNA structure. So here's how the probabilities are calculated for the Gibbs sampling step. So given these 2 sequences, what I want to know is the probability that position I and J in sequence A are paired to each other, and also that positions K and L in sequence B are paired to each other, and furthermore that AI is aligned with BK. So I'm simultaneously asking that they pair to each other and they align to each other, because I'm trying to do a multiple alignment and structure prediction simultaneously. And so I'm going to just multiply all those probabilities together. I don't have time to go into a whole lot of details, but the idea is you start with this initial information, and then you do a sampling thing. We're sampling based on stems, so we're going to pull out stems and then rank them based on these probabilities of what's the probability that these 2 stems from these 2 sequences are aligned with each other, and that they have they're comparable stems. And from this collection of stems, you can put them together into structures, and then you can go back and you can update the probabilities. And so you just do this, and it doesn't take very long to converge. So we applied it. We had a grant from DOE to work on Chihuanella. They had sequenced about, 15 genomes or something. And so we took those genomes. We specifically looked at the regions around the AUGs, and we found a number. These are a bunch of predicted motifs, RNA structural motifs, and many of them correspond to things we know about. They weren't known about in Shewanella before, but there could have been picked up by lots of programs. Many of them you could find just by looking for terminators because they correspond to leader sequences, for example. Some of these were found by qRNA, some were not. Some of them correspond to, I forget, one of these on here is actually known to be a riboswitch in another species, the tRNA synthetase gene. So a number of these we're pretty confident are real. So anyway, we have a method that we think works. This is actually an active area of research, so a lot of other people are working on it as well. But mostly just telling you that it's a problem that I haven't entirely abandoned. I've still been working on it to some extent. But what I want to do now is go to the specific point about post-transcriptional autoregulation. And I'm going to just make sort of a list of a few examples. Some of these are classic, well-known ones. So ribosomal RNA proteins, certainly in bacteria, but also it's known to occur at least sometimes in eukaryotes, are autoregulatory. Their primary target is they bind to the ribosomal RNA, and as soon as the ribosomal RNA is saturated and you make more of that protein, what does it do? It binds its own messenger RNA and turns itself off. The messenger RNA has evolved to look like the ribosomal RNA target of that protein. There are several examples of tRNA synthetase genes as well. So their primary target is the tRNA, and they put the amino acid on it, and when that is saturated, when they've finished their job, the next place they bind is their own messenger RNA to turn themselves off. I'm going to show you two examples that I think are really cute. There are some splicing factors that are known to be autoregulated. In polycistronic messenger RNAs, and this really relates to this ribosomal proteins right here, you really only have to turn off the first gene in a polycistronic message because all the rest of them are coupled. So again, they're regulated via internal RNA structures. So this one, we knew about this a long time. In fact, we actually wrote a paper about IF3 proposing a mechanism. It turned out that mechanism was wrong, but I can tell you about the story anyway, because it's a really, I think, interesting story. So this is, first of all, RF2 is a release factor 2 from E. coli. It recognizes the UGA stop codon. So when a ribosome comes to UGA stop codon, it releases the ribosome. Well, it turns out when it was first sequenced, it was discovered that it has a UGA stop codon right in the middle of its own gene, right? So what that means is that if there's plenty of this protein around, it terminates its own synthesis. But when you're low on that protein, when there isn't very much ribosomal of RF2 around, what happens is the ribosome gets here, it stalls, it pauses, and then it reinitiates out of frame. And it continues, and so you make the protein. So it's a very nice way. So in this case, the protein hasn't had to evolve at all. It just does its normal function. The messenger RNA has evolved to take advantage of that function to do autoregulation. IF3 is initiation factor. Its function is to help select the initiation codon to bind to the AUG. It will allow GUGs and other things, but it's pretty strict. If you in vitro, if you leave it out, then you get much more prolific translation at non-AUG start codons. Well, it turns out its own start codon is an AUU. I think it's the only example, but that's its start codon. So the idea is that when there's plenty of IF3 around, it basically doesn't translate itself. But whenever IF3 falls to low levels, now it starts translating at other start codons, including AUU. So again, the protein hasn't evolved, it just does its normal job. The messenger RNA has evolved to take advantage of that for autoregulation. Here, I mentioned RNA coupling. This is another project that I happen to get involved in. So the gene here, this is the AUG and the Shiner-Delgarno for the lysozyme gene of T4, which you want to turn on late, right? After the phage has gone through and done everything, then it turns on the lysozyme gene to basically burst out of the cell. You don't want to turn it on early. Well, it was discovered that the message is made early. It's just not translated early. And once we sequenced it, or we didn't do it, but once it was sequenced, it became clear why. There's this strong hairpin. What I'm showing you here in the dotted lines, those are the late promoters. So late in infection, it makes a new messenger RNA that doesn't have this structure, and therefore it makes lysozyme late. But the model here is that there's something, there's the upstream gene, which is the hydrolase. There's something that controls this, and under some conditions, if this gene gets expressed, then you also express this one. Now that's, as far as I know, it's still a mystery. It's not clear how this upstream gene is, but it's a way that you can take some gene upstream, you regulate its translation, and once its translation gets activated, then you would activate this one early. It's be as an escape mechanism, so the phage could get out with a low level.
Want to summarize?
Yep. Okay. I'm sorry.
It's great.
I'm sorry. I lost track of time, and
It's perfect.
Okay, let me just tell you about one last example, and this is one of my favorite. It's the first project I did as a postdoc. Gene 32 protein in, again from T4, is a single-stranded binding protein, single-stranded DNA. It's like SSB and E. coli. It turned out that Larry's lab showed that it was auto-regulated at the level of translation. Pete von Hippel's lab had done all the characteristics. They knew what the binding affinity was to different sequences. It was basically non-specific, but they also showed that it bound to DNA much better than RNA. It turns out the way it regulates its own expression is its first job is to saturate all the single-stranded DNA in the cell, and then the next thing it does is it starts binding to RNA. Well, its own RNA target is the first place it binds, and the reason is because it's a very cooperative protein. The cooperativity factor is 200. So what it does is its first messenger RNA that it binds to is its own messenger RNA. Here's the sequence. Okay, so that was another example. Let me just jump to the conclusion here. So I think that in general you can have some gene X. You can have, when its activity is high, its expression is low, and when its activity is low, its expression is high. All it means is that somehow the protein, its expression is coupled to its activity. And again, it can be done in a way where the protein itself doesn't have to go through any evolution. It's only RNA that has to evolve and then interact with the protein in some way. I think there's likely to be common just because there are many opportunities for post-transcriptional regulation. Structural motifs are harder to find. Here's the point I want to get to. Autoregulation, it provides a really efficient way to do coupling. You really can determine the amount of a protein pretty accurately by controlling it at the level of translation. It's easy to evolve. Messenger RNAs can evolve to bind proteins. You can always find an RNA that will bind to essentially any protein, so you can imagine that messenger RNAs evolve in such a way that they get activated whenever the protein is in excess of what's needed. Now, I don't know how often this happens, but it's plausible, and I suspect it happens a lot more than we know about. As a challenge to synthetic biologists, you could design a regulatory system entirely of post-transcriptional autoregulation, like the kind I was talking. You basically make aptamers to a protein, and you tune them to the right KD, so whenever the protein level gets above that KD, it binds and turns off its expression, and whenever it goes below that, then expression happens. Okay, I have to give just some acknowledgments. These are the current people or recent people in my lab who did the Motif Discovery stuff I talked about at the end. These is a list of old collaborators, and you'll see many familiar names on here. I've been very, very fortunate in my career to have some really fantastic collaborators. I know you recognize lots of these names. And our current collaborators on the Motif at the experimental side are Scott Wolf, Michael Brodsky, and Keith Young. Here's a picture with some of the people, and here's the final sculpture of my lab done by an undergraduate in my lab. So I apologize for taking over. I'll be happy to answer questions. Nobody's going to challenge me on my speculations. Everybody believes it or everybody knows it's BS.
A quick comment. Please let us know where you're standing when you ask a question, because it can be difficult to tell in this
Right-hand side?
I hear somebody.
Yes, here.
Oh, yes, okay.
You mentioned protein motifs in the context of DNA binding, but have you applied these methods for just protein motifs?
Protein motifs. You mean proteins within the motifs within the proteins themselves?
Yeah, for protein interactions or for specific enzyme.
We haven't done that. Other people have done that. Sort of. I mean, in the late 80s, people started building, profiles, and there's these PFAM things, and sometimes those are long, but sometimes they can be small motifs that correspond to, like I didn't show you, but you have a zinc finger motif, for instance, which is 30. So there are those kinds of motifs that people discover and model by similar methods. But we haven't done very much of that ourselves.
Thanks.
Yes?
All right. I have a question. Have you ever looked into, or do you know anything about different mRNAs having preferences towards ribosomes composed with different rRNA genes? Because there is a cluster of genes, right, which are all a little bit different from each other. Some of them are differentially expressed, so some variants of rRNA are tissue-specific. What about interaction between, different mRNAs and different rRNAs? Is anything known about it?
Not by me. An interesting thing. I mean, most of my work in post-transcriptional regulation has been at the prokaryotic level, and there, even when they have multiple ribosomal RNAs, they're usually identical or nearly so. So I'm not actually familiar with the idea of having multiple different classes of ribosomal RNAs. It's interesting, but I don't know anything about it.
Yeah, I mean, I think most of people think that they are identical, but or there is some small variation that is not relevant to anything. But there were a few papers recently showing that there are tissue-specific rRNA genes. So.
Oh, that's interesting. I didn't know that. Yeah.
Hi, Gary. Nice talk.
Thanks.
It's an okay body of work, I guess.
What's that?
Sorry, I was just joking. Thanks for highlighting our work on motifs for RNA binding proteins. Just to say, we actually did report structures that were preferred by those proteins if we could detect a preference. And the only reason I bring this up is right immediately after that, you started talking about finding long stems that are conserved. And this comes up a lot when people talk about RNA binding proteins, and certainly the RNA binding proteins that we have motifs for, they're not going to be recognizing those motifs within the context of long stems because we're not going to be able to find them.
Right.
And a lot of people the first thing they try to do is they try to model these complex structures that are recognized by RNA binding proteins, and we've never really seen evidence that most of the RNA binding proteins that we have domains for recognize these kind of complex structures. So just as...
Yeah, no, I think you're right. And I mean, I just went over your slide. I think it's a really nice paper. I just flashed it up there to say this is a lot of interesting work going on in this field. And I think you're right, but mostly what you're working on are these things that have RRB domains and things like that. And I think you're right. As far as I know, they tend to recognize either unstructured things or short little motifs. But I'm also thinking about, and maybe these are not the classic RRB kind of proteins you're talking about. I didn't show pictures of it, but things like the ribosomal proteins that they recognize, for instance, there's a really great example from David Draper of this very complicated pseudoknot structure that it recognizes. And there are other ones like that. The tRNA synthetases that control their own expression. What happens is their messenger RNA has evolved to look like a tRNA, and it has, in fact, all the loops, everything that is needed for recognition. So there are examples of very complex structures recognized by proteins, but most of the time that I know of, at least, those are kind of one-off things where that protein recognizes that thing only. And the kind of things you're looking at are sort of like transcription factors. You have a protein that binds to lots of motifs all over the genome, and I suspect in those cases they are going to be these sort of shorter motifs, lower information content. Maybe they have structure, maybe they don't. And they'll be sort of more like transcription factors in that way. But I think there will be also cases where things bind to really complex structures. Maybe they'll be less frequent.
I totally agree. Thanks for clarifying that.
Sure.
One comment and one question.
Okay.
To my knowledge, the first DNA-based Helix type of work was done, it's published in Nucleic Acid Research, and the technique is called target detection assay. It was published in 1990. So I'm one of the authors. I'm the inventor.
Okay.
And now a question to the...
That was for a DNA binding?
It was DNA binding, yeah. Nucleic acid research, and the idea I had back in '87
yeah.
at the EMBL in Heidelberg.
Yeah, I think I do remember that paper, actually. I mean, there's a list of papers that came out sort of the same time. Actually, Kevin Struhl did something very similar. It didn't have a PCR step involved, but it was actually in '89. Oliphant and Struhl had something that was very similar to cell
It's true, but it's true, but... Right. But the DNA work with the PCA, yep. with oligonucleotides and PCA right. steps and amplification, that was target detection assay, TDA assay, nucleic acid research, 1990.
Yeah, okay.
But the question now is, I'm highly excited about your work on your zinc fingers. So can you find the CTCF sites in your in vitro binding assays? Or selection?
We've done that I mean, all of our in vitro assays are done at 2 fingers at a time. We wanted to, I mean, some of the early work was actually just done by single fingers, but we knew that there was a context dependence on adjacent fingers. So all of our discovery thing, and the picture I showed were all 6 long binding sites corresponding to 2 fingers. So that's all of our assays were based on that. But then you can take those and you can stitch them together and you can make predictions of proteins that have any number of fingers. And we've done that, compared it to some CellX data from different groups. Now what happens is, as the protein has more and more fingers, what happens is typically you don't get all of them. Sometimes you get a few of them, and it may be that those proteins, they don't use all their fingers for all their recognition, or if they have that many fingers, only some of them matter very much or whatnot. For CTCF, as I recall, that's been a complicated one. I can't remember right now how well we did on that. In this paper we're writing now, we have a whole bunch of things where we compare our predictions to what's known about some of these proteins with lots of fingers. And the predictions aren't as good as they are on our
Okay.
cross-validation, but I can't answer your question.
Yes, back to the history. In my paper on zinc finger cloning of human zinc fingers in 1990, I came up with the expression of whether there's a zinc finger specific recognition code. So I'm not aware of somebody else who at that time was discussing this issue.
Okay, great. Let's talk about this later. We should probably wrap up, I think.
So let me pause the discussion in the interest of time and thank Gary Stormo again for a fantastic speech. Thank you very much. We have Birds of Feather and student session, student council career session. Remember your tickets to dinner tonight.